continuing on with relation to the questions which have been coming in Karnataka entrance. The next possible questions that have been coming in right through from the last 10 years include rDNA enzymes. Now, when you say rDNA enzymes, R refers to recombinant DNA enzymes. That is, enzymes which help in joining DNA pieces like ligases, enzymes which are termed as NICs, which can break down nitrogen bases, NICs, as well as we have helicases, which can dismantle the sort of DNA, they can separate the sort of DNA, ligases, and again we have even DNA polymerase. Now, DNA polymerase enzymes are those which go to form the polymer. So, DNA polymerase enzymes are very important enzymes which start the sort of a formation of the DNA during DNA replication. Now, when you take DNA polymerase could be of type 1, type 2, as well as type 3. In addition to this, we also have primase. Primase are special enzymes which are present in cells which act like a primer. They act like a primer, that is, they are the start enzymes. So, primarily a good knowledge of all the recombinant DNA enzymes which are involved in DNA replication, which are involved in transcription, as well as in translation, and in the central dogma. Form essential readings. So, while when we are doing the objectives, we would be checking on the different sort of recombinant enzymes and the sort of rules. The second cluster of questions can also be coming from, so the first is rDNA enzymes, second cluster can come from Agrobacterium tumefaciens. Agrobacterium tumefaciens is a tumor inducing bacteria, soil, rod negative bacteria, and primarily Agrobacterium tumefaciens induces crown gall disease, and the crown gall disease induction is due to the presence of tumor inducing principle or the TI plasmid. The tumor inducing plasmid predominantly has been one of the most important discoveries in the 1980s, and we understand that Agrobacterium tumefaciens is a natural genetic engineer. Because it can directly take in an exogenous DNA, an outside DNA, and integrate it into the plant host. Third series of questions which normally can come in is, what are GMOs? When we take GMOs, genetically modified organisms, there are several sort of probable questions which are coming in. One of those includes Dolly the sheep. Then we also have Bacillus thuringiensis, BT plants. Among BT plants, we have BT brinjal, which has the cry gene, which is resistant to aphids. Then lot of questions come from golden rice. Golden rice is transgenic rice. Transgenic rice, whereby rice is fortified. It is fortified with vitamin A. and thereby this is also an example of genetically modified foods and also genetically modified organisms. So when you take three clusters, recombinant DNA technology, agrobacterium tumefaciens, as well as genetically modified organisms, and also we have the concept of the golden rice, which is very, very important in objective bits, and students should also be knowing about what is the Bacillus thuringiensis plants and how does Bacillus thuringiensis act.